Namaste and welcome back to Typical Monk. Last class, we learned the tools and steps of biotechnology. Today we see where all of it is actually used, from Bt cotton in the field to insulin in a vial. And we will drill it the neat way too, one idea at a time. Here is today's path. 5 stops. First, Bt crops, that is, pest-resistant plants. Second, RNA interference for pest resistance. Third, genetically engineered insulin called Humulin. Fourth, gene therapy and molecular diagnosis. And fifth, NEET drills, that is, assertion, match, and statement. Let's start in the field with Bt crops. The bacterium Bacillus thuringiensis makes a protein crystal that is toxic to certain insects. The genes are called Cry genes. Chi1ac and Chi2ab protect cotton from bollworms. Chi1ab guards corn from the corn borer. Inside the bacterium, the toxin is an inactive protoxin. When an insect eats it, the alkaline gut dissolves the crystal and activates the toxin. The active toxin binds the midgut lining, makes pores, the cells swell and burst, and the insect dies. Now, a clever way to make a plant fight a pest. RNA interference. RNA means RNA interference, a natural cellular defense that silences a specific messenger RNA. The tobacco plant was protected from the nematode melodorogene incognita using this method. Nematode genes were introduced by Agrobacterium, making both sense and antisense RNA that pair up into double-stranded RNA. This double-stranded RNA silences the parasite's own messenger RNA so the nematode cannot survive in the transgenic plant. Now, a life-saving medicine, genetically engineered insulin. Insulin is 2 chains, A and B, joined together by disulfide bridges. The body first makes proinsulin, which has an extra stretch called the C peptide. This is cut out to give mature insulin. In 1983, the company Eli Lilly put the A and B chain sequences into E. coli plasmids, making each chain separately. The 2 chains were then extracted and joined by disulfide bonds to form human insulin, called Humulin. Now, can we fix a faulty gene itself? That is gene therapy. Gene therapy means correcting a defect by delivering a normal gene into a person's cells or tissues. The first attempt, in 1990, treated a 4-year-old girl with ADA deficiency. The enzyme adenosine deaminase was missing. Her lymphocytes were grown outside the body, given a functional ADA gene by a retroviral vector, and returned to her blood. Because these cells are not immortal, the treatment has to be repeated. It is not yet a permanent cure. Now, how do we catch disease early? That is molecular diagnosis. Ordinary tests act late. Early detection needs to find the pathogen or the faulty gene, even at very low amounts. PR amplifies tiny amounts of DNA. It is used to detect HIV in suspected AIDS patients and gene mutations in suspected cancer. ELISA works on antigen and antibody interaction. It detects the pathogen's antigen or the antibodies made against it. Together, PR and ELISA catch an infection before symptoms appear, and that is the goal of molecular diagnosis. Now, the high-yield pointers, the facts that NEET turns into questions. 1. Bt toxin comes from Bacillus thuringiensis. Its genes are Cry genes, and the toxin is stored as an inactive protoxin. 2. The Bt toxin is activated by the insect's alkaline gut, and it kills by making pores in the midgut cells. 3. RNA silences a specific messenger RNA using double-stranded RNA used against the nematode melodorogene incognita. 4. Insulin has chains A and B plus a removed C peptide. The product is Humulin, made in E. coli. 5. The first gene therapy treated ADA deficiency in 1990, and PR and ELISA are the molecular diagnostic tools. Now the first drill, assertion and reason. Each time, ask, is it true? Is R true? And does R explain A? First, A, the Bt protein does not kill the bacillus that makes it. R, the Bt toxin exists as an inactive protoxin, and is activated only in the insect's alkaline gut. Both are true, and R correctly explains A. So the answer is A. Second, a genetically engineered insulin is made as a ready two-chain molecule inside E. coli. R. Bacteria assemble the C peptide better than humans do. Both statements are false, so the answer is D. Third, R. Aided deficiency gene therapy must be repeated periodically. R. The corrected lymphocytes are not immortal. Both are true, and R explains A, so the answer is A. Now, the second drill, match the column. Make the correct pairing. Cry genes. The Bt toxin against insects. RNA interference, nematode resistance. Humulin, insulin from E. coli. And ELISA, antigen and antibody detection. Now the third drill. How many of these statements are correct? Check them one by one. First, the Bt toxin is activated in the insect's alkaline gut. This is correct. Second, gene therapy for aided deficiency gave a permanent cure in 1990. This is wrong. Third, RNA interference silences a target messenger RNA using double-stranded RNA. This is correct. So the correct statements, 2, that is, the first and the third. Now let's clear the mistakes students fall for. Myth: Bt cotton is toxic to every insect that merely touches it. Truth: the toxin acts only after it is eaten, in the alkaline gut of specific insects. Myth: insulin comes out of E. coli as a finished molecule. Truth: the A and B chains are made separately, then joined by disulfide bonds. Now test yourself. Answer each one before it is revealed. Answer first. That is how you build strong NEET recall. Question 1. The source organism of Bt toxin. Answer: Bacillus thuringiensis. Question 2. Which pest was controlled by NI in tobacco? Melodogene incognita. Question 3. The host used to make Humulin insulin, E. coli. Question 4. The first disorder treated by gene therapy? ADA deficiency. Question 5. The technique that amplifies tiny amounts of DNA? Pisama, Isha. Let's gather it all in one breath. Bt crops carry cry genes. The protoxin is activated in the insect's alkaline gut. RA use S double-stranded RNA to silence a pest gene, giving nematode resistance in tobacco. Humulin, the insulin A and B chains made in E. coli and joined by disulfide bonds. Gene therapy first treated ADA deficiency in 1990. Repeated and not yet permanent. And PR and ELISA give early molecular diagnosis of disease. In the next class, the ethics of biotechnology, DMOs, biopiracy, and patents. See you there. You have now seen biotechnology at work, feeding crops, curing disease, and detecting illness early. Be proud, one idea at a time, with a calm mind, and you will crack NEET. This is Typical Monk. Every class, always free.